Assalamualaikum and hi everyone. My name is Nurul Anna Fatihah binti Azhimi and I will be presenting on my final year project entitled Garlic and Chili as the bio-insecticide against ants in UiTM Cawangan, Pahang. My supervisor is Dr. Muzamil bin Mustafa. These are the contents of this presentation. Background of the study. Ants are social insects that can pose property and health risk. Thus, to overcome ants infestation, chemical insecticides often used for immediate insect killer. However, most chemical insecticides contain harmful chemicals that can cause adverse health effects. Natural products are claimed to exhibit comparable efficacy to the chemical insecticide, so garlic and chili are often used as biological insecticide on a wide variety of insect pests. However, there was not much research on effectiveness of garlic and chili against ants. The problem statement was there were insufficient studies on phytochemical screening on garlic and chili for insecticidal properties. Furthermore, the studies on efficacy of garlic and chili on ants was also insufficient. Other than that, biological insecticides are unpopular to be used for household. The significance of this research is to develop an eco-friendly and cost-effective insecticide. The development of biological insecticides also will provide new knowledge to the public to develop their own natural insecticides, thus can reduce health risk and environmental impact. The first objective of this study was to determine the phytochemical properties of garlic and chili. The second objective was to determine the insecticidal activity of garlic and chili extracts, and the third objective was to compare the insecticidal activity of biological insecticide with the commercial insecticide. Past research found that the phytochemical screening on garlic and chili showed the presence of alkaloids, flavonoids, polyphenols, saponins, and tannins. Alkaloids act as growth inhibitors and feed restrictions, while flavonoids can inhibit the juvenile hormone formation, the excretion in metamorphosis, and reproduction of insects. Polyphenols display a feeding deterrent in herbivorous insects. Saponins can reduce the food intake and disrupt the activity inside the insect's gut due to the toxicity and reduced digestibility. Tannins also reduce digestibility associated with the undigested feedstuff. Garlic and chili claim to exhibit insecticidal activity against common pests. Past research on garlic juice against maize weevil showed that the Nigerian garlic cultivar juice exhibited total mortality compared to the German garlic cultivar due to the concentration of allicin. Next, the research on ethanolic chili extracts exhibited highest larval mortality compared to the acetone and crude extract of chili due to the different bioactive compounds extracted. Then, the combination of garlic and chili extracts exhibited highest mean yield of tomato fruit per hectare due to the reduction of red spider mites population on the plant. For comparing the efficacy between commercial and biological insecticides, it was found that the Veratrin, a botanical insecticide, was comparable with the abamectin as it exhibit slightly lower density reduction of spider mite. For the first objective, the determination of phytochemical properties of garlic and chili was done by identifying alkaloids, flavonoids, polyphenols, saponins, and tannins through several chemical tests on the powdered material. For the second objective, the determination of insecticidal activity was done by preparing the garlic, chili, and combination of garlic and chili extracts and test on the ant that was collected in Tok Gajah College. Mortality test was done in petri dish containing filter paper that was soaked with treatments, and a total of 5 ants were transferred into the petri dish and were observed for every 5 minutes for 30 minutes. No movement of antenna or curling of the body will indicate the mortality of ants. For the third objective, the comparison between biological insecticide and the commercial insecticide was done using supplementary, and the mortality test method was the same as in objective 2. All data obtained were analyzed using one-way ANOVA followed with Tukey post hoc test. The p-value under 0.05 is considered significant. The result of the first objective indicated that A. sativum and C. frutescens does not exhibit presence of flavonoid based on table 4.1. Ethyl acetate supposedly extract flavonoids from the sample, as flavonoids are semipolar compounds that can be extracted by semipolar solvent. The past study showed that the fresh garlic ethyl acetate extract has higher TFC compared to the isopropanol extract. Other studies also suggest that Chilli ethyl acetate extract has higher TFC compared to N-hexane, chloroform, and ethanol extract. However, the study by Ahmed and Noamo showed low concentration of flavonoids in garlic compared to other compounds, which was taken into consideration for the present study. Not only that, but polyphenols supposedly present in garlic. Past studies showed that the TPC is higher in black garlic alkaloids extract at 0.25 gram per ml compared to at 0.125 gram per ml. From here, it can be said that the concentration of extract plays the role in TPC level. The result on the second objective indicated that the test ant to be used was Monomorium pharaonis. The worker ants are monomorphic with varying body size of 1.5 to 2 mm long. They displayed body color of golden yellow to reddish brown. Based on table 4.2, there was statistically significant difference between the treatments as determined by one-way ANOVA. Two key post-hoc tests revealed the mortality of ants statistically significantly different when both C-fructosans and the combined extracts were applied compared to the A-sativum extract. However, there was no significant difference between C-fructosans with the combined extracts. A-sativum had statistically significant increment in ants mortality after 25 minutes compared to after 5 minutes. The combination of A-sativum and C-fructosans showed statistically significant increment in ants mortality after 25 minutes compared to 5 minutes. C-fructosans extract exhibited highest mean number of ants mortality, which is 3.33 at 0.1 gram per ml by 30 minutes. It was followed by the combination of A-sativum and C-fructosans, which is 3.00, and A-sativum, which is 1.67. This results contradicts with the past study where the combined extract supposedly exhibit higher mortality compared to the single extract. In that study, the combined extract attained 100% mortality of bedbugs within 10 seconds at 10 mg per ml compared to the chili extract with 23 seconds, and the least effective was the garlic extract with the 300 seconds. However, if compared only between chili and garlic extracts, it is similar to the past research where chili is more effective compared to garlic extract. For the third objective, the comparison between biological and commercial insecticide was presented in table 4.3. From table 4.3, there was a statistically significant difference between treatments as determined by one-way ANOVA. The two key post-hoc tests revealed the mortality of ants was statistically significantly different when A-sativum, C-fructosans, and the combined extracts were applied compared to cypermethrin solution. Cypermethrin exhibit statistically significant increment in ants mortality by 15 minutes compared to by 5 minutes. Cypermethrin solution exhibited highest mean mortality of ants, which is 5.00 at 0.001 gram per ml by 33 minutes compared to C-fructosans, which is 3.33 at 0.1 gram per ml. Effectiveness of C-fructosans extract somewhat comparable to cypermethrin solution. In the past studies, cypermethrin exhibit highest reduction in infestation of plant shoot by 4th week of treatment compared to the 1st week after treatment. It was followed by neem oil that exhibit slightly lower reduction at 5% concentration level at the 4th week. The least effective was A-sativum extract with the lowest percentage reduction at 4th week. In conclusion, for determination of phytochemical properties of garlic and chili, the phytochemical screening for garlic exhibited the presence of alkaloid and saponin, while chili displayed the presence of alkaloid, polyphenol, saponin, and tannin. For determination of insecticidal activity of the extracts, the test ant species was identified to be Monomorium pharaonis and was used to test the insecticidal activity of the extracts. The application of chili extract exhibited highest mean number mortality of ants, which was 3.33 at 0.1 gram per ml, among other extracts within 30 minutes. For comparisons, the efficacy of extracts was compared with the cypermethrin solution. The application of cypermethrin solution exhibited highest mean number of mortality of ants, which was 5.00 at 0.001 gram per ml within 30 seconds. It can be said that the chili extract efficacy was almost comparable to that of cypermethrin. Recommendation: chili has the potential to become a safe and efficient household biological insecticide. However, further research is needed to clarify the insecticidal activity of chili extract against a wide range of insect pests, and the active ingredients in the extract that are responsible for insecticidal activity on ants should be identified. These are the selected references used in the presentation. Thank you for watching. If you have any questions, kindly contact me through email or phone number displayed on the screen.